to invite you all. Good afternoon, everyone from Dar es Salaam, Tanzania. Welcome to the Environmental Biology, Chemistry, and Toxicological Online Journal Club presentation. As Dr. Kojo has indicated, our presentation today is on dietary acetate promotes growth and the nutrient deposition in inert tilapia through increasing acetyl coenzyme A triggered energy production. As I have been introduced, Samuel Mchere Rimbu in the Department of Aquaculture Technology, School of Aquatic Sciences and Fisheries Technology of the University of Dar es Salaam, in Dar es Salaam, Tanzania. This work was done in collaboration with Laboratory of Aquaculture, Nutrition and Environmental Health in the Department of Biology, School of Life Sciences at East China Normal University, Shanghai, China. Just briefly, that the University of Dar es Salaam and the East China Normal University established a joint research center for aquaculture and fish biology. It is through this collaboration where we are able to conduct this study. I will now dwell into the presentation. My presentation is divided into 5 sections, as shown here on this slide. I will start directly with the introduction on something on here. I want to pull it up. It is distracting me. Yes, the physiological function of short chain fatty acids. Something is not working. Okay, the short chain fatty acids are produced by some microorganisms in our intestine through the dietary fiber that we consume. And of course, the main short chain fatty acids which are known include acetate, propionate, as well as butyrate. And of the 3 short chain fatty acids, at least acetate is the main dominant type of short chain fatty acid. The intestinal short chain fatty acids, they participate in several physiological functions in different tissues, and so they regulate some of the metabolism in our bodies, in the immunocells, move in the bone marrow, in the liver, in the brain. You have some of them functioning for some time. And so the intestinal short chain are distributed almost throughout the tissues during blood circulation for regulating different metabolic activities in our bodies. However, the 3 short chain fatty acids, they have different metabolic characteristics. We all know that the short chain fatty acids are all involved in energy production. However, propionate and the butyrate, they usually require mitochondrial beta oxidation in order to produce acetyl coenzyme A, which participates in the TCA cycle for energy production. On the contrary, the acetate, which is the dominant one, does not involve the barrel oxidation, but it enters more rapidly as a direct substrate for acetyl enzyme A production for the different functions within the TCA cycle. Now, the question is, what are the physiological functions that are performed by acetate in fish as one of the organisms? The last several physiological functions which are performed by acetate, and as a short chain fatty acid, acetate is also produced in the intestinal fish guts from non-digestible complex substrates which the fish consume. So the gut microbiota, through some fermentation processes, they usually produce the acetate as one of the short chain fat acids. The short chain acids have been established in different species, such as zebrafish, as it was in the previous slide when I showed that acetate is usually the dominant one. Here you can also see that the content of acetate is significantly higher than that of propionate as well as butyrate. Equally, in United tilapia, the intestinal short chain fatty acid was also dominated by acetate as opposed to the other two types of short chain acids. Now, acetate as the dominant short chain fatty acid have been subjected to different studies in order to understand the physiological function it performs in fish. Actually, in zebrafish, dietary sodium acetate enhanced the feeding and improved energy metabolism. You can see here the daily feeding rate was increased significantly in the fish that were fed acetate as opposed to the control diet. Equally, the energy here was significantly higher in the fish that were fed on acetate as opposed to the control diet, which at the end of the day increased the body weight of the fish. Similarly, in Nile tilapia, dietary sodium acetate improved metabolic disorders caused by high carbohydrate diet through acetate supplementation. In another study, dietary acetate also enhanced the immune system in Nile tilapia. Now, these studies indicated that acetate improves various physiological functions in fish. Now, the big question is, what is the effect of acetate as a substrate for energy source on the systematic metabolism in fish? Now, this was our main question which we asked for this start. To answer this question, we designed an experiment which was in vivo. And for this start, we used the 39 tilapia weighing 5 grams, at least in 3 replicates. So we made a control diet. And then, of course, we added sodium acetate to the control diet in order to make the diet which we called sodium acetate diet. Now, this diet was fed to the two groups of fish, the control as well as the sodium acetate for a period of 2 months. We did it for 8 weeks. Now, at the end of the start, we analyzed acetate, glucose, lipid, protein, as well as energy metabolism in the body of the fish. And we mainly focused on the liver as the Center for Metabolism in Organism. Our aim here was to conduct a systematic evaluation of all the metabolic effects when acetate is used in fish. Now, what did we get after conducting this study? We started by establishing the metabolism of acetate in the fish. We did this because we know that we are sure that the acetate metabolism is mainly conducted by using two genes, the acetate coenzyme system 1 as well as system 2. So we wanted to know what might have happened to these fish within the intestine after giving them with a diet that was composed of acetate. So the acetyl coenzyme synthetase short chain 1 as well as 2 was analyzed to see what happened in the bottom of the fish. So we found that the protein level expression of the two genes, at least in the fish that were fed on the acetate diet, were significantly increased, as can be seen here, as compared to the fish that were fed on the control diet. However, feeding the fish with the control diet and the sodium acetate did not affect the relative acetate content in the river. Interestingly, feeding the fish with the acetate increased the content of acetyl coenzyme A as opposed to those that were fed on the control diet. Now, in general here, these results show that dietary sodium acetate increased intracellular acetyl coenzyme A content through the acetyl coenzyme A synthetase short chain 1 and 2. This was very interesting to us. So we asked ourselves, if the intracellular acetyl coenzyme A was increased, what could then happen to the glucose metabolism? So we analyzed the effect of sodium acetate on liver glucose metabolism in the Nile tilapia. We started by conducting a glucose tolerance test, where the results indicated that the Nile tilapia that were fed on the sodium acetate had a quicker return to the baseline serum glucose as opposed to those that were fed on the control diet. However, for the insulin, it was relative. The fish were resistant to insulin changes as opposed to those that were fed on the control diet. We further went on analyzing some genes which are involved in glucose metabolism. We found that most genes involved in glucose catabolism, such as glucokinase, pyruvate kinase, peptidase dehydrogenase, as well as peptidase K, were significantly inhibited in the fish that were fed on the sodium acetate. Interestingly, the fish that were fed on sodium acetate increased the glucose transport gene, GLUT2, as well as glycogen synthetase gene. We further analyzed some metabolites in the glucose, in the glycolysis process, where we found that the fish that were fed on sodium acetate reduced significantly glucose 6-phosphate, as compared to those were fed on the control diet. However, pyruvic acid was significantly increased in the fish that were fed on the sodium acetate diet. Moreover, because we saw that the glycogen synthesis gene was increased, we wanted to confirm if glycogen in the liver was increased. Indeed, we found that the fish which were fed on the sodium acetate diet had a significantly higher glycogen content as opposed to those that were fed on the control diet. Now, all these results indicated that feeding the fish with sodium acetate, it actually inhibited the glucose catabolism, but promoted glycogen synthesis. This was quite interesting to us because we know that glucose is particularly catabolism is involved in energy production. So we then asked ourselves, if glucose, particularly catabolism, was inhibited, what could happen to the lipid metabolism? So we went further by analyzing the effect of sodium acetate on the liver lipid metabolism. Here, we started by analyzing some genes that are involved in lipid metabolism. And we did this at messenger level as well as at protein level, where we found that the genes that are involved in lipid catabolism, such as CPT1A, as well as adenosine triglycerol-lipase, were significantly inhibited in the fish that were fed on lipase, both at messenger RNA expression level as well as at the protein level. We further checked on the hepatic autophagy to see if it was also affected. Here we found that most genes that are involved in the autophagy, such as becrin, as well as LOC3 and the P62, were significantly increased in the fish that were fed on the sodium acetate diet, as opposed to those that were fed on the control diet. We assumed that something might have happened in form of lipid accumulation. Therefore, we analyzed the liver TG as well as non-esterified fatty acids, which we confirmed that they are both elevated in the fish that were fed on the sodium acetate as opposed to those that were fed on the control. The lipid accumulation was also confirmed through histology, on which the fish that were fed on the sodium acetate has more lipid droplets as opposed to those which were fed on the control diet, as you can see here. Therefore, in general, on lipid metabolism, as we saw to the glucose metabolism, it was clear that feeding the fish with sodium acetate inhibited lipid catabolism in order to increase hepatic lipid accumulation in the body. So far, the glucose catabolism as well as lipid catabolism were both inhibited, which are important in the energy production. We then asked ourselves, now, if these two were inhibited, what could happen to the last macronutrient that is used in energy production? So we analyzed the effect of dietary sodium acetate on liver protein metabolism in the body of the fish. We started by looking on some amino acid metabolism, where we found some interesting story here that the serine protein kinase gene, as well as the activating transcription factor 4, as well as glutatase, were significantly inhibited in the fish that were fed on sodium acetate, as opposed to those that were fed on the controlled diet. Another thing that was inhibited was the ketone-based gene, the branched chain keto alpha acid dehydrogenase was also significantly inhibited in the body of the fish. We analyzed some free amino acids within the liver, where we found several of them, including arginine, histidine, lysine, methionine, tryptophan, as well as valine and glutamine. All these were affected. Some of them were increased, while others, like this one here, tryptophan, was decreased in the fish. And we wanted now to confirm if protein synthesis was affected. We did this by analyzing the mechanistic target of lampamycin, which is known to be involved in protein synthesis, both at phosphorylated and the total mTOR were analyzed in our stats. As can be seen here, the mTOR was significantly enhanced in the fish that were fed on sodium acetate, as opposed to those that were fed on the control diet. In total, these results here on the protein metabolism again indicated that feeding the fish with sodium acetate actually inhibited the amino acid catabolism, but it activated the protein synthesis. This was a very interesting result to us. If you sum up all the three macronutrients, the glucose, the lipid, as well as the protein, the catabolism were inhibited. Now, we then asked ourselves, what could these fish get the energy from if the catabolism were inhibited? So to answer this question, we then analyzed the effect of acetate on the liver energy metabolism in the fish. Of course, we know that the mitochondrial tricarboxylic acid cycle, as well as the oxidative phosphorylation pathway, are two important processes which are involved in energy production in the living organism. So we started by analyzing some of the metabolites which are produced within the TCA cycle, taking into consideration that the lipid, glucose, and the protein metabolism, protein catabolism were all inhibited in the fish. After analysis, we found that citric acid, just somewhere here, the aconistic acid, as well as the NAD and NADH, somewhere here, as well as the citrus synthesis gene, again on this area here, were all significantly inhibited in the fish. On the other hand, we found that oxoglutaric acid, as well as the succinic acid and the isocitrate dehydrogenase, these genes were significantly enhanced in the body of the fish within the TCA cycle. We went further to analyze the metabolites within the mitochondrial oxidative phosphorylation, where we found that the cytochrome B2 and adenosine triphosphatase gene, as well as ATP, were all increased. These metabolites were all increased in the fish that were fed on the sodium acetate diet, as opposed to those which were fed on the control diet. Now, generally, if you look on these studies, on these results, you will see that the sodium acetate actually promoted the acetyl-CoA energy production through mitochondrial TCA, as well as the oxidative phosphorylation pathway. So since the lipid, glucose, and protein catabolism were inhibited, the fish relied on the acetyl-CoA to produce the energy by using the TCA cycle, as well as the oxidative phosphorylation system. We wanted to confirm more on the effect of the acetyl-CoA on energy production by analyzing some more metabolites that are involved in acetyl-CoA. And to do this, we analyzed the overall protein acylation, as well as the mitochondrial membrane protein acylation levels. Interestingly, all these were increased in the fish that were fed on sodium acetate, as opposed to those that were fed on the control diet. Moreover, we also analyzed acetylcarnitine, as well as ketone body, on which we found that the fish that were fed on the sodium acetate increased the levels of acetylcarnitine, as well as ketone body, highlighting that actually the exogenous acetate altered the intracellular acetyl coenzyme metabolic pathway through acetate by inhibiting some of the catabolism, the amino acids, the lipid catabolism, and of course influence the several metabolites that are involved in the TCA cycle. So all these results indicated that sodium acetate actually increased the metabolites when acetyl was used as a substrate. Now, the fish had limited catabolism, but they used the energy from the acetyl-CoA. We then asked ourselves, what could this mean to the general physiological function of the fish? So we analyzed some growth performance and the other related parameters in the world of the fish, where we found that the Nile tilapia that were fed on the sodium acetate increased weight gain, body length, carcass ratio, conditioning factor, and of course it deposited more protein as well as lipid in their body. We further analyzed liver inflammation, antioxidant apoptosis, and some liver health, where we found that most of the genes that are related to inflammation, such as toll-like receptor 2, NF kappa beta, the tumor necrosis factor alpha, as well as tumor growth, transforming growth factor beta, were significantly inhibited in the fish that were fed on the sodium acetate, as opposed to those that were fed on the control diet. For antioxidant capacity, we found significantly higher superoxide distributors, as well as catalase in the fish that were fed on the sodium acetate as opposed to the control diet. The apoptosis, we confirmed it by using the TAN, such that the fish which were fed on the sodium acetate actually had reduced apoptosis in the hepatocyte. You can see here the lead is relatively not shown, not clear here, but here you have them more vivid, indicating that apoptosis was reduced in the fish that were fed on the sodium acetate diet. For the liver health, we clearly show that the aspartate transphanasigine content was significantly reduced in the fish that were fed on the sodium acetate, as opposed to those that were fed on the control diet. Now, what does these results mean? In general, they show that the sodium acetate increased nutrient deposition. As we can see here, protein and the lipid content were increased, but also it improved the liver health. You can see here the AST is significantly reduced in the fish, which promoted the general growth of the fish that we saw. Now, generally, what do all these results mean to us? In summary, what we have seen is that the dietary derived acetate was effectively converted into acetyl-CoA through the acetyl-CoA short chain system, which affected most of the metabolic processes that we saw. That is the first take-home message. The second one is that the sodium acetate actually promotes growth and the macronutrients. We saw the protein and lipid were all increased in the body of the fish. And of course, they improved the liver health of the fish. And lastly, the dietary sodium acetate activated the mTOR signaling, which is known to affect protein synthesis, and simultaneously inhibited the catabolism of the three main macronutrients, and therefore it promoted the energy formulation from the acetyl-CoA rather than from the three macronutrients that are known to provide the energy. Lastly, I wish to thank the National Natural Science Fund of China for funding this study. And of course, I'm also happy and thank you to my two collaborators in China, Professor Jane Yudu, as well as Professor Zhang Meilin, and all the laboratory members at the Laboratory of Aquaculture, Nutrition and Environmental Health for their cooperation during this study. I would also not forget the members from the School of Aquatic Sciences and Fisheries Technology of the University of Dar es Salaam for the collaboration on this work. So for those interested in this work, as I have shown in the group, they can just download it through the Journal of Aquaculture, which we managed to publish it. Thank you very much for your attention. That is the end of my presentation. Thank you.
Thank you very much, Dr. Samuel. I now understand why you're not having the time to review this paper earlier on. There was a lot of work that was done on this publication. So we would like to straight away delve into the next section of the review. The third one, reviewer. If you are here, please can you unmute yourself and then give us a quick synopsis of the paper, and then we can move into the Q&A section. Thank you. One of our colleagues volunteered to give a synopsis of this paper. Are you here? If you are here, unfortunately, I don't know your name, so I can't call you by your name. Can you unmute yourself and then give us your assessment quickly, and then we can move into the forum where you did a Q&A session.
Hello. Sorry, please,
Yes.
can you repeat that?
Yes, there was one of us who volunteered to give an assessment, a quick synopsis of the paper as a co-reviewer. If you are here, please unmute yourself and do that quickly, and then we can move into the Q&A session.
Well, Dr. Haneyo asked me to review the paper, to be a lead reviewer on the paper. Am I the person you're looking for?
I'm sure you are the person, sure.
What did you say, sir?
Yes, I think you are the.
Hello?
Hi, Doc, carry on. You are the one I'm looking for.
Hello?
I can hear you. Can you hear me?
Yes, I can hear you now. You went off a little, you went mute again. Okay,
Okay,
so hello? You're going on mute and unmute. So yeah, I think I'm the one you're looking for then. Dr. Haneyo asked that I be a lead reviewer on the paper, on our discussion, just to lead our discussion with all other colleagues on the work done and presented by Dr. Samuel.
Yes, please carry on.
Okay. Thank you very much, Dr. Limbo. It was a very, very nice presentation, and I really enjoyed it. I actually enjoyed reading the paper, and a lot of questions that I had, prior to your presentation, some of those things have been clarified. So some of my questions, some of my comments, go in line with questions in terms of clarifying some details of your methodology. Interestingly, I've been looking at the chat box and I've seen, one or two questions that I've also identified here, especially one that I see from Dr. Salisu. So I'm going to launch in right away. Just like the moderator said, it's a lot of work that you have done. It's a very, very beautiful study. So, and to appreciate it better, it would be nice to, have a little bit more of clarity on what was done specifically. So I'll start from the materials and methods section. And my first question is, okay, in the design of the experiment, okay, there was aspect of acclimatizing the fish for a period of about 2 weeks, feeding them on a commercial diet. So it's kind of like a question. And then after the acclimatization phase, you then translated into feeding them with your own kind of prepared fish feed formulation. I'd like to find out why you had to transit from that to designing your own specific fish feed, and then it is that same fish feed, based on my understanding of what you explained there, that you then augmented, supplemented with the NACC, with the sodium acetate supplement. So my first question here is, why did we not just simply, supplement the commercially available diets? Why was it necessary for you to actually prepare your own, fish feed technically? And then it would have been interesting to compare these results from your own direct fish feed, okay, which you formulated, your own formulation that was augmented or supplemented with the acetate, vis-a-vis the commercial, commercial diet, which was used to acclimatize the feed at the beginning of the experiment. So I would like to actually understand the rationale as for why that kind of setup was done. Then the second question still on that is, okay, so what informed doing the experiment at 8 weeks? Because the fish feed you set up in 6 tanks, 30-30 in triplicates. So that means, by my understanding, the one that was not supplemented, that was just your formulated fish feed alone, you had 30-30 fishes in each tank in triplicates, which is a total of 90 fishes. And then you had the second setup in triplicates of the formulated fish supplemented acetate in another 30-30-30 in each tank. So I would like to know, why 8 weeks? Why did we terminate the experiment at 8 weeks specifically? Is it because maybe at that particular point in time, that's the best, what do you call it now? Best time to assess some of the parameters that you assess? Is that the maximum age at which, the fish grows, and maybe that's at that point in time that it is harvested and consumed? So I would like to know the rationale for terminating the experiment at 8 weeks. Now, so I got a little bit confused when, in the section 2.3 of the paper, where you assessed, where you said you took 12 fishes. Okay, so I would like a little bit of clarification in terms of what exactly was done. So did you pick four fish from each triplicate, making total of 12 for the experiment itself? So that means you assessed 12 for the control, which in this case is your formulated diet, and 12, which is for the formulated diet with the acetate supplementation. Because the impression this gives in the paper is that you harvested a total of just 12 fishes. Okay, so I just wanted to clarify that my understanding was correct. Now, a little bit of going now into the, what do you call it now? The qPCR experiment. I wanted to find out what informed the use of 2 different internal references, because I happen to do a lot of qPCR experiments as well. And usually we use just one internal reference. I mean, because the idea of using the internal reference for comparative gene expression analysis is to be able to calculate relative to the expression of a gene that is constantly expressed, that you're sure is going to be there, instead of going into absolute quantitation. So I would like to know what informed the choice of those 2 internal references. Though it's not detailed in this paper, what was the difference? Did you find any difference in the relative gene expression values when you calculated or determined using beta-actin or the elongation factor 1? Because those were the 2 that you used. So I expect that in the determination of the relative expression, you definitely have calculated or determined the expression of each of them individually, and then informed that in determining your mean expression, comparative gene expression value. So I would like to know, okay, was there any significant difference in, the gene expression values when you looked at both internal references? So the last part of my, then I can now go into some of my other questions. Like I said, a lot of them were sort of like answered in the course of the presentation. So to look at, the overall work itself, I found it interesting that there was generally reduced catabolism and there was increased anabolism. So basically, you are building up, making the acetate treatment increased, the production of proteins. Of course, that was how do you say it now? That was evident in the fact that there was increased protein content. Okay, so it's quite interesting. So I'm kind of like wondering, at what point does this become how do I say, at what point does this become a disadvantage to the plant? Because you're just building up and building up, there's no breakdown. And of course, we know that in biological living systems, the balance of buildup, which is anabolism and catabolism, has to be maintained. At what point, and this is where the issue of 8 weeks becomes interesting to me. I know that we breed these fishes for consumption. So I'm just interested in the overall well-being of that fish. At what point does this balance, this tilt in the balance? Yes, you saw that there was no significant effect on the overall health of the liver, which is very, very interesting because the liver is not stressed at that point. So you're going through buildup. At what point does it tilt back, to the catabolism pathway? So I'm interested in knowing, okay, how that dynamics work vis-a-vis, this build-up mechanism, promotion of acetyl-CoA synthesis, of acetyl-CoA build-up in the cell, as well as build-up of some of these other beneficial amino acids and lipids. Particularly the lipids worry me because you know that over time, if lipid builds up in the system, it could ultimately have a negative effect on circulation, eventually in the blood of the organism. So I'm interested to have your thoughts on what could happen, relative to this. So I kind of like your explanation, your suggestion about why you had increased protein activity for the ACS. We had increased activity for the ASCCI 1 and 2 gene proteins. We had a reduced expression of those genes, and then we had an increased concentration of the acetyl-CoA, which is the product of that pathway. So I liked your explanation because that actually is what makes a lot of sense, that could be as a result of, a post-transcriptional or translational, most likely post-transcriptional modification of that gene, and which is regulated itself by the sodium acetate. So for me, I would want to know at what point, is that tilt? What do you think? What are your thoughts? At what point would that tilt? Because yes, you're having increased, okay, the sodium acetate seems to be regulating, the expression of the gene. You're having high activity of the protein, and then you're having high acetyl-CoA production. At what point, do you think the delicate balance of that pathway, that regulation, may tilt? And then, or is it also documented in literature that sodium acetate could build up of sodium acetate in the system? Does it have any negative effects on the overall well-being of the organism? Okay, so this is why I'm saying this. There was a work I was involved in where we were trying to study the pathway of thiamine biosynthesis. And we found that the active product of thiamine biosynthesis, which is thiamine diphosphate, is actually dangerous in the blood. So thiamine diphosphate tightly regulates, the presence of thiamine diphosphate could shut down the entire system because it's not supposed to build up in the blood, which is why I'm asking the question relative to, does sodium acetate in the system, does it have anything to do with that overall pathway of, the acetyl-CoA accumulation vis-a-vis the expression of the genes and proteins proteins that are involved in the pathway of breaking down the sodium acetate that is fed to the fish? I don't know. Okay, so maybe I also have another question, which a little bit you clarified, and I would like to know more about this. So you talked about the fact that the acetate was a preferred SCFA compared to the propionate and the butyrate. So I'm just out of curiosity, could this be an issue of bioavailability while we have more of the acetate available in the system vis-a-vis these other two forms of SCFAs, which is the propionate and the butyrate? Do you think it has something to do with bioavailability? You already said that the propionates and the butyrate have to be, beta oxidized before in the mitochondria. Before then, you can actually get them to become available. But I'm just interested to know, because this is your area of expertise. Does this have any effect on bioavailability? Yes, you need to go through an extra layer or extra step, you know, in terms of breaking down the propionate or the butyrate. Does it have any implication on bioavailability? And for future directions in terms of studies, I would maybe be interested, you know, it's just something that came to my mind. Okay, what about, you know, combination of these SCFAs, that is vis-a-vis maybe acetate, propionate together as supplemented by acetate, butyrate, you know, to see if those two could, you know, added into the diet could maybe have any effect, you know, on the energy metabolism, of course, the yield and the overall health of the fish. So that is something that, you know, comes into mind in terms of future directions. And another quick question that I have to ask is relative to the ratio. What ratio of the acetate, okay, because you formulated the diet, in the overall diet, what percentage did the sodium acetate in the overall diet that you prepared? Okay, because I see in some of the literature that you reviewed where it says that, okay, 0.18% of the dietary sodium acetate, okay, alleviated some certain symptoms in the fish in previous studies. So what percentage, what proportion of the diet was the sodium acetate? Yes, I know you said it was 1,800 grams per kg that you use, but what proportion of the diet is the sodium acetate, you know, in the supplementation? So those are the few questions and some of my thoughts relative to the work. It's a beautiful work. Thank you very much.
Hello.
Hello.
Kojo, should I go directly to answer these questions, or are there any other modalities?
Yes, Doc. It can respond to the questions step by step as you went through them.
Okay.
Thank you.
Okay, thank you very much. Fadipe? Was that Dr. Fadipe?
Yes, please. That's me, sir.
Yeah, thank you very much for the questions. I have noted about 7 questions that you asked. The first one was related to acclimatization and feed formulation. And
yes,
you asked it specifically, why did we had to make another diet rather than using the commercial diet?
Yes, sir. Yes, sir.
Yeah, okay. So yes, you could opt to use the commercial diet. However, you know that during feed formulation, anything that is done, it has to add up to 100%. So the commercial diet that you are using has actually a formulation of up to 100%. That means there is no room to add more nutrients, meaning that at least, because whenever we formulate, we usually assume that the formulation is per kilo of the diet. So if you have already attained the kilo, there is no empty space to put some more ingredients in your formulation. So the commercial diet has its own formulation that is that does not allow addition of the sodium acetate that we wanted to include in our formulation. That is one. But also Usually for experimental studies, you want to have pure nutrients, the nutrients which you are really sure of the quality, such that if there is an effect in the body of the fish, you can see it clearly. So these two reasons made us make it our own feed rather than using the commercial diet. The second question is, if I got it correct, was on the time of the experiment.
Yes,
where
sir.
you indicated that we did it for 8 weeks. Well, this is sufficient. Yes. Unfortunately, this is a common practice in aquaculture, that whenever we do some experimental studies in aquaculture, particularly for the experiment aspect, we usually do it for 8 weeks. And the reason here is clear, that we usually target that the fish should double their initial weight. When the fish have doubled their initial weight, any effect, particularly to the fish farmer, will be clear, because the fish farmers are looking to produce bigger fish in the shortest possible period. So when the weight of the fish has doubled, you are sure that the effect of the treatment will be clear in the body of the fish. If there is no effect, it will be clear that, oh, nothing has happened with my treatment. If there is an effect, you will vividly and clearly see it, because at least the weight of the fish has doubled. So based on the growth performance of the fish, the 8 weeks are enough to double the weight of the fish, where you can see clearly the differences in your treatment. The third one was on the 12 fish, which I should just confirm with you that yes, the way how you thought, 4 times 3 fish was our, so for each treatment, we had 12 fish, because we had 3 replicates. Then we sampled the 4 fish from each, and we just got the 12 fish. For the internal references, we used the 2 internal references with just a simple reason. We wanted to be sure that we get accurate results. In one of our studies, we had a lot of comments from one of the reviewers indicating that only a single internal reference sometimes might not be enough. So to avoid this, in this study, and of course in several other studies, we used the 2 internal references. You further asked on this if there are any significant differences. Indeed, there were no significant differences, but just some minor differences between the 2 internal references that we used. So at the end of the day, our results were calculated based on the average of the 2 internal references. In this way, you get the accurate results, which has no any other queries for someone to think that probably the internal reference was not correct. And then you had the increase of acetyl vis-a-vis the Derik's balance between catabolism and anabolism. Unfortunately, I would say that we didn't dwell too much on this, of which probably the issues which you are mentioning might be correct, because we did not go into details on this aspect to see where is the Derik's balance between the catabolism and the anabolism, and at what level. Really, on this aspect, we did not go into details to analyze. On the negative effect, based on our results, if you see the inflammation and the liver health, we could not find, we found that the liver health was actually improved rather than affected. So probably we might need to do some more studies on this aspect to see if there are still some hidden negative effect on using this sodium acetate. But at least for our results, particularly based on the AST activity, as well as some of the inflammatory genes, you see that the liver was really improved because both AST and some of the genes, including the tumor necrosis factor, were significantly reduced, indicating that the liver was in good health as opposed to those fed on the control diet. The acetate bioavailability. I wanted to think that it is, we know that the number of carbon atoms for acetate and the other two components, the other two short chain fatty acids, they are different. We think probably it is based, the dominance of the acetate might be related to its carbon, but I'm really not sure on this, whether it is its bioavailability is higher or rather because it contains only two carbons, probably it is easier for it to be involved in many metabolic processes such that whenever you measure, you find its concentration is higher as opposed to the other two short chain fatty acids.
Doctor, can I ask, can I just say something relative to this? So I was trying to understand that, okay, if it is definitely the propionate and butyrate, since the active form of this short chain fatty acid in the system happens to be the acetate form. So even if at some point you even feed the animal with a propionate form or the butyrate form, you should always find an accumulation of the acetate form because you're going to definitely need to first break down those other two forms. So you should expect to maybe find them reduced because they are not the active form. It seems the acetate is the active form that goes directly into the acetyl-CoA. Of course, that is broken down into acetyl-CoA and gets into the energy metabolism pathway. So I'm thinking that this may not be an issue of the fact that, okay, it's not an issue of they are not really beneficial. That's those two forms. It's just that maybe there's a step of, having to be broken down first to release the acetate form that goes into the pathway and then is broken down to acetyl-CoA, which is why you always find an accumulation of acetate. What do you think?
Rather than the propionate. I would
exactly.
think it might be the way how you are putting it. Although, as you indicated, probably, if we can do a start feeding the fish with the 3 different types, probably singly and in combination, we could get a very interesting story on what actually happens within the body of the fish.
Of the fish, yeah. Very, very good, yeah. Okay, please continue, sir. Thank you.
Okay, the last one, if I recorded them correctly, was on the proportion of sodium acetate
In the diet.
in the diet. Yes, as we indicated that we used 1,800 milligram per kilogram acetate, and this was actually confirmed from our previous study as the optimum level required to cause some effect in the fish. So here, the 1,800, they are milligram per kilogram. That means if you are interested in looking for a ratio, is simply you can convert this 1,800 milligram into gram, such that you can get in a kilo of the diet, because whatever formulation we do, we usually do it per kilo of the diet. So you could see if this 1.8%, all other 18, if my calculations are correct here, it's like 18% of the diet. If you are making 1.8, I think 1.8% in percentage wise. So the 1,800, they are milligram in 1 kilo of the diet. So you can go further to look for the ratios or anything that you wanted to express it. But as you are making the diet, at least just make 1,800 milligram into a kilo of your diet. As I said, we confirmed this in our previous 2 studies, Rieta 2022, as well as Shueta 2020. If I can read them somewhere here.
So
2022. Yes.
Yeah, I saw that in the study that you did to arrive at that weight. But you know, in your literature review, your introduction, you talked about the different percentages that have been done in literature relative to zebrafish, which was, you know, fed with 0.5% of sodium acetate. You talked about 0.18% of acetate also, especially for the Nile tilapia, which is your work, you know, that alleviated lipid accumulation and intestinal inflammation. And, you know, I actually pointed out that I was worried about the accumulation of the lipids that were being made, that was not being broken down. So, which was why I was interested in knowing what percentage was yours in your diet at the end of the day. Was it higher than this 0.18% that was used in the previous study?
This should
that you mentioned. This was one of the things that sort of like stimulated my interest in knowing what actual percentage of the diet was the sodium acetate that you incorporated into the fish feed, so that one could maybe understand or maybe do a better comparison of the work that was done, or which on this particular acetate that alleviated, you know, the issues of hepatic lipid accumulation and inflammation. Because at some point, like I said, these lipids that are being accumulated, that is being made, that is not being actively broken down, would get to a point where it might begin to impair the health of the fish. Of course, you said at 8 weeks, it didn't have this effect. But what if at a longer time, you know, it affects the health of the fish?
Yeah, that's why studies are usually there. And of course, probably this was an experimental study. So for commercial users, then you have to conduct a study on a field where the fish could be fed from the very first days until when they are harvested. As you were talking, I was trying to convert the 1,800 milligram per kilogram into percentage, and I see here is 18%.
It's 18%.
Yes.
So it's definitely like times almost 1,000 more, 100 times more than what was, you know, used in the study that saw the alleviation symptoms of lipid accumulation in the previous work that you quoted. So it kind of makes sense that it's,
Yeah.
you are having,
Yes.
lipid accumulation at the end of the day. Okay. So
Yeah, yeah.
one thing is, I know that this kind of study that you've done eventually is supposed to translate into the field where, the fish farmers will adopt this type of fish formulation, I mean, they are interested in the yield. They want it to, the fish to be bigger. Of course, you want it to be also more nutritious. It's kind of good that we're having a buildup of this beneficial stuff in the fish. And so when we consume the fish, we get our nutrients from it. It's interesting on this perspective of for the fish farmer at the end of the day. Oh, my fish is rich in protein, my fish is rich in this, is rich in that, which is good. So, but I just wanted to know, at what age is this fish harvested in the field and usually is sold? Okay, because if it goes beyond probably 8 weeks, which you've done in your study, could it be that by the time the fishes are harvested and being sold, which is for the profit of the farmer, we could have gotten to the point where some of this accumulation that is not, that is making more anabolism, less catabolism, could have become, detrimental. So the age at which the fishes are harvested and sold, I think, would be maybe an important indices for people, or for fish farmers that want to sell, for profit.
Yeah, if I can comment on that, we are lucky that, particularly my collaborators in China, whenever we do some studies, I also did mine, we have one of the feed manufacturers whom we collaborate with. And this feed manufacturer have an experimental setup such that when we do the studies at experimental level, we then have to take the same into the field. Now, from the field studies, we can now be sure of the total effects of a certain additive that we want to introduce into the industry. Therefore, for this kind of, for sodium acetate, we are still doing some more on-field studies with the feed manufacturer, such that if we are sure beyond the doubt that what happened at the experimental level will happen to the fish in the tanks, in the ponds, then the technology will go to the farmers. Now, on the issue of the size of the fish, usually Nile tilapia harvesting, it takes like 100 grams to 300 to 500, and this is approximately 6 months. You will have the size of this kind of fish, but this is very much dependent on your consumers. If there are consumers who are willing to buy even less than that, then some farmers usually harvest them earlier. But at least 200 to 350 up to 500 is the appropriate size and the common in the markets.
Yeah.
Yes.
So I am kind of happy that this is something that is definitely, you are doing this in collaboration with the fish farmers themselves, as much as we're interested in the commercial aspect, the profitability aspect. We also have to think about the overall health of the fish, vis-a-vis by the time you're harvesting it and all of that, because I'm really worried, I must tell you, about those lipids that are building up really inside the liver of the fish, and then it's not being broken down. So it will be interesting. Maybe, I don't know if you could, in further studies, also increase, beyond 8 weeks, and then see maybe, specifically, maybe some of these parameters are altered down the line. And of course, you know, it's interesting, a lot of future work direction that could be done. It's a great work. Congratulations for this kind of work. Good job to your team, sir. And
Thank
it's
you
been
very
nice
much.
talking to you and discussing this paper with you. I hope most of the participants have found something interesting from our conversation in terms of the review of the paper. And some of my comments have been helpful for you. I hope they've stimulated future directions for you, for your work.
Yes, thank you very much for your insight, your questions and thinking. Yeah, this is how science should be. You produce and then the people ponder about that and they see it at different angles to see what can be improved. Thank you very much for your comments and suggestions.
Thank you very much. Thank you, sir. Thank you. Yes, sir.
Thank you.
All right. Thank you very much, biochemist. You took me to school to a course that I hated so much when I was in my undergrad, biochemistry. But notwithstanding, this is the application. And thank you very much for that interactive section. I would like to open the forum. If we have questions, we have 5 minutes today that we have exceeded our time. So our plea that will be very snappy and so we can leave here for those of you who have other things to attend to do so. Please, if you have a question, open your mic. There are quite a number of comments and questions in the chat. I think that Samuel has done that to most of them. However, I will go through it to the outstanding ones. I will verbalize it. If you have a question, please raise your hand. Dr. Salisi, please, the floor is yours.
All right, thank you very much. Thank you, Dr. Kojo. Yeah, we are learning. I'm happy. Thank you, sir, for the presentation. I won't take your time. Thank you so much, Dr. Kadipe and other participants for learning together.
Yes.
I am just interested in the formulation. I am presently working on fish feed and I would like to learn more. So we can, I'm also looking forward to formulation of fish meal in my area that can enhance most importantly, the health of the fish now. Because like you said, most of the commercial fish meal that we have here, they proclaim to have a particular percentage of protein, which is not the case. So that is why I'm actually interested in the formulation report. And I would also like to know if there's a journal for your university where we can publish. Thank you.
Thank you, Dr. Salisi. That was very brief. Dr. Samuel, would you like to respond to the comment? I think she ended with a question and a journal to publish together.
Yes, I would. I appreciate Dr. Salisu for the comment. And of course, the formulation, probably if you write, we can even communicate after this meeting to get more details on the formations. But for the journal, yes, we do have the journals at the university. There are several of them, but of course, related to this, we have one journal known as Tanzania Journal of Science, which has some of this kind of work within it. But as you might be aware that probably because of our policies, sometimes we don't publish more in our local journals. We target some international journals in certain publishers like Elsevier, which we think, of course, they are more reputable than whatever we have here. But at least we have the Tanzania Journal of Science and the other journals, depending on the field which you publish, actually do some publication, publish some articles in different areas. The university has several fields, and basically at the moment, we have almost a journal for every field that is taught at the University of Dar es Salaam. So if you are interested, you can submit your next paper to some of our journals. And the Tanzania Journal of Science, which I mentioned, I'm actually an editor within that journal. I just forgot to include it in the short brief that was given to you. Thank you.
Thank you very much, Dr. Lindu. If there are no hands, let me go to the chat. I think there was a question by Dr. Benga Olude. If you are here, can you unmute your mic? I think they have quite a number of questions.
Thank you very much. It was quite a great work. I think some of those things Dr. Samuel has talked to them, and I feel, because I didn't quite read the publication before joining in, so when the reviewer, Dr. Fadupe, was reviewing, I got to know that some of those things I was asking for are there. But also, I want to provide clarification. Dr. Samuel was talking about the level of the sodium acetate being 18%. You know, it couldn't have been 18%, 1,800 milligram out of a kg. I tried to provide, you know, the calculation there. And more so, 18%, that's going to be a lot. You know, if 18% of your diet is going to be an additive, you know, that's going to be a lot. That is just what I want to say for now. Most likely, you know, maybe I will be contacting Dr. Samuel, you know, for collaboration in the future. Thank you very much.
Thank you very much. That is
If I can, Dr. Kojo, if I can say something on that.
Sure.
Thank you very much, Olude, for doing the calculation. As you can see here, I'm listening, answering, and trying to do the calculation. So I think I got it long, and it is true that it is 0.180%. It's very, very little amount as you did the calculation. So I was calculating while listening and trying to answer. I think I added 100 more than the required level. So it has to be 0.18%. Thank you.
All right. Thank you very much. From the look of things, I think we have exhausted all our questions and comments. I would like to thank you, Dr. Samuel, for making the time out of your busy schedule to present the detailed study. I'll continue to classify as a very detailed study. At the moment I'm in China, and I know how we design our studies, a very detailed study. And congratulations to you for this. And then he mentioned something that I think I would like to emphasize to all of us, especially to our colleagues back home who have students we are working with, doing a couple of researches in your various institutions. Here in China, what I realized is almost every lab have an industrial partner. For instance, my lab has like 3 different industrial partners, and every research that we do, we have to make sure that it is tested on the real field that can be commercially useful to the society. And so I think it's sort of a model that we need to adopt as Africans, especially with our various institutions. Just like you mentioned, we have a commercial partner that took the result and also tested it in real life to see how this formulation could be useful to feed seed that could be used by fish farmers. So I think it's something that with our research institution, we do not even have the funding. I mean, these entrepreneurs such as Canon, they are looking for you and I who have the brains, who have the knowledge, who have the experience to be able to explain the science behind what you're doing. Just approach them. Some of them will just need you to engage with them and they'll come on board. So I think we really need to emphasize in our research as we are starting off in our continent. One of the solutions to the problems of our continent is research. And we can't be doing that at the academic level only. Let's engage closely with the industry. just like the Chinese are doing it. There's nothing that we do here that goes with every experiment we do to have an industrial partner. So I would like us to glean 1 or 2 lessons from this and see how we can adapt this in our various institutions back home in Africa. I'd like to thank you all for making time to participate in this section. I'd like to thank Dr. Samuel once again. I also like to thank our lead reviewer, Dr. Tenito. That was a whole school of biochemistry you took me through again. And like I told you, I never liked biochemistry when I was back when I was in school. Thank you for making it real and making it more interesting for us this afternoon. And thank you all for participating. Thanks my colleague Ravi Chandra for coming through and also giving your support behind the scenes to make this successful. Dr. Samuel, my last request to you, we would like to publish this on the Author Aid blog. And so we need your input. I will draft something and share with you to make it, and then we can publish it on the website of the Author Aid blog. So thank you very much. We wish you a very restful weekend. And so we'll meet again for our next session. Thank you. I'm signing off from here. Thank you everyone for making it.
Thank you.